The reef wakes before the sun clears the swell. A single large ruler patrols a living fortress, every fin stroke a reminder of order. Then absence. She is gone, vanished into the blue. By morning, the script resumes with a different lead. Bodies alter, voices drop or sharpen, pair bonds shuffle. Eggs become sperm, or sperm become eggs, as chemistry drafts a new chain of command. Stability returns, yet something fundamental has moved. This is animal behavior recast in minutes and days, not generations. Call it animal behavior, how sex switches on schedule, written in color, posture, and charge. Huddled within the tentacles of a venomous host, ocellaris clownfish, Amphiprion ocellaris, build small societies that rarely stray far. One anemone, one territory, a queue of bodies graded by size. The largest is female, the next largest is the breeding male, and below them a handful of non-breeders held in check by threat and ritual. Adults usually reach 6 to 11 centimeters, 2.4 to 4.3 inches. They feed on plankton near the anemone and nip away parasites from their host. Reproduction runs on a calendar of lunar light, temperature, and nest tending. The male fans eggs and guards the clutch. Here, though, the headline is stranger than caretaking. Animal behavior, how sex switches on. Schedule describes not a rare accident, but a life plan etched into anatomy. Sequential hermaphroditism, specifically male to female transition under social control. The trigger is a social jolt with physiological teeth. Remove the dominant female and the breeding male begins to transform. Neural circuits in the brain change activity. Gonads reorganize from testis to ovary. Courtship displays rewrite themselves. Timelines matter. Behavioral dominance can shift within hours. Gonadal restructuring often accelerates across days, while full ovarian function, vitellogenesis, ovulation, and fertile spawning emerges within weeks. During this window, the hierarchy reknits. The largest non-breeder ascends to breeding male, keeping the pipeline of larvae intact and the anemone defended. The costs are real. While the transition proceeds, gamete output stalls and territorial pressure rises. Yet the system holds, which brings the paradox into perfect focus. What hormonal and social cues trigger rapid sex change, and why is the switch adaptive for territorial reef life? The question cuts through the coral like a cold front, because everything here depends on timing, neighbors, and one immobile address. Evidence suggests two cascades intertwine: social perception and endocrine pivot. Visual and chemical absence of the female lifts suppression on the breeding male. Aggression from subordinates tests his resolve, but the queue remains orderly. Size policing enforces the ladder. Inside, steroid profiles flip. Studies indicate a surge in brain and gonadal aromatase activity, the enzyme that converts androgens to estrogens. Circulating estradiol climbs. 11 keto-testosterone falls. Genes tied to ovarian differentiation, including CIP19A1 and FOXL2, ramp up, while testis-associated markers recede. The hypothalamic pituitary gonadal axis responds. GnRH pulses adjust. Gonadotropins tune the gonad. Even the laryngeal voice, pops and clicks used in disputes, shifts as tissues remodel. Scientists still debate whether cortisol, the stress hormone, serves as a master switch or a background modifier. Either way, the social vacancy writes straight into biochemistry. Why hazard a midlife rewrite? One hypothesis draws on the size advantage model. In these fish, female fecundity scales steeply with body size. Larger females produce many more eggs. Male success, in contrast, depends less on size because a single male can fertilize a large clutch. The strategy becomes clear. Mature first as a smaller male, then switch to a bigger, more fecund female when the vacancy opens. That sequence maximizes lifetime reproduction across the group. The trade-off is downtime during transition and vulnerability to rivals, but the long-term yield is higher. A second hypothesis emphasizes territorial economics. Each group lives on a single anemone, a scarce and defended address. New recruits seldom arrive. Suitable hosts may be meters apart, yet effectively worlds away. If a breeding female disappears, importing a replacement is unlikely. The quickest route to restored reproduction is local conversion. The social cue ensures the largest, most experienced resident steps into the role. This is adaptation scaled to a postcode, not a coastline. A third lens looks at conflict management. By enforcing strict size ranks and holding non-breeders in stasis, the group reduces costly fights. When the top roll opens, everyone moves one rung. The system limits chaos and preserves the anemone's defense while keeping reproduction near continuous. Still, this order has limits. Prolonged environmental stress can delay transitions. Small groups risk collapse if multiple adults are lost. These bounds matter because animal behavior is not a script so much as a negotiation with chance. Together, the models outline why a switch, seemingly impossible to explain at first glance, is workable, even efficient, in a territory that cannot move. Night cools the reef, and the anemone folds slightly, as if to breathe. The new female settles heavier among the tentacles, and the former subordinate tests his new post by fanning an empty patch of rock. Within days, eggs will appear, amber beads under his care. The cast has changed. The story has not. We watch for a signal, a note that starts the conversion, and meet a silence that hums with chemistry. Nobody knows why one lineage perfected this timetable while neighbors did not. But the result keeps rewriting what we thought we knew. Animal behavior, how sex switches on. Schedule lingers here in the space where a vanished ruler becomes a hormone, a gene, a new order, then a clutch that proves the wager paid.